Welcome back, and let's start now with term 3 topics. This is subtopic 6.2, that's Mendelian genetics. First of all, 6.2.2, relate using examples between genes and alleles. So we need to talk about like what are genes and what are alleles. So of course, every one of you know that gene, a short segment of DNA that codes for a specific protein. This is how you define the gene and how we go for the alleles. So, and it's written over here, alleles are alternative forms of a gene. Okay, so this is the definition of the allele. Which of the following best describes the relationship between genes and alleles? So genes are alternative forms of alleles. No, we already said this is the definition of allele. Genes and alleles are the same. No, alleles are found only in the no. So B is the answer. Explain how geneticists use the principles of probability. Probability is what? Chance of an event to occur. So this is how we say that it's a chance of an event to occur. We need to go for the laws of probability. There are two laws of probability. If there is an event happening and that event A and B are mutually exclusive, like this is either one or the other, like, so how you will say it as probability of A plus probability of B. But if A and B are independent, like mutually exclusive means they come together or they go together, but in case of independent, it's like either head or tail, and this is the most commonly used probability rule in the Mendelian genetics. Using a Punnett square, which of the following answers predicts the probability of obtaining a recessive offspring from a cross between two heterozygous parents, and explain why? Okay, so recessive offspring, you know that when you have two heterozygous or two carrier, okay, they look normal. So for example, capital A crossed with capital A small a crossed with capital A small a. So when you do the Punnett square for this one, so it will be like this: capital small and small. So this is the one that you need. So it is 25%. So 25%, we have only one answer, and because each parent can contribute either capital A or the small a because they are independent. This is how it happens. Which of the following applies when the probabilities are multiplied? So we already said when the probabilities are multiplied, the events are independent of each other. So this is how it works, independent. So when how you will say it then when the events are independent? When they are mutually exclusive, when it is mutually exclusive, you will say it as probability of A plus probability of B. This is how you will say. You will use plus rather than sign, rather than the multiply. Which of the following applies when the probabilities are multiplied? Probabilities are multiplied, but it's being used over here as a plus. So when the probabilities are multiplied, I think it should not be multiplied. It should be over here when the probabilities are what they were using. When the probabilities are added, it should be here added. This is wrong question. So when the probabilities are mutually exclusive. Now imagine if the probability of an event to happen, for example this one, the probability of an event to happen is 1/4, and if this is going to use this rule, so it will be 1/4 crossed with 1/4. So this is how it will come if the same event happen another time. So in the first time you have 1/4 probability, and in another time you again have 1/4 probability. The probability never change. It's same as, for example, there is a couple, and this couple has already 3 boys, okay, and 2 girls. Now they are expecting another baby. So what will be the probability? Okay, so most of the students, they answer as a girl because there is one less girl. No, but that's not the case. It's not hard to fast rule, so it's going to be either boy or a girl. That's 1 upon 2, 1 upon 2. So how you will calculate the probability of a boy to girl? So 1 over 2 upon 1 over 2. So this is how you will calculate the probability, because both events are taking place as 50-50. Describe pedigrees and their components. So first of all, you should know like what is a pedigree. So pedigree is a diagram to represent the transmission or the inheritance transmission of a trait in human. So in human, we don't make crosses. We normally represent it with the help of a pedigree, and it's like a diagram. How can a pedigree be used to determine whether the trait is dominant or recessive? So you can easily see over the generations. You do check the pattern of the inheritance across the generations. So by measuring the frequency of the trait, no. By examining the physical characteristic, no. By analyzing the pattern, yes. So this is the answer. By conducting genetic tests, no. Genetic tests we only do for certain special cases. It's not like always being done. So be very careful when you are answering. Okay, let's move to the next word. Label different parts of the pedigree below. So we need to label different parts. So this is a pedigree. Be very careful when you are gonna answer it. So if it is a circle, it's a female. If it is a square, it's a male. Open symbol means it is not affected, and dark symbol mean it is affected. It works for both, like this one as a male and the other one as a female. Now the line that is between male and a female, this line, this is a horizontal line between them, and this represents mating. Then after that we have the line that is coming from these parents downwards, like this one. This is a vertical line that leads to the progeny. Progeny mean offsprings. Progenies now are arranged horizontally left to right. So it starts from here and goes to that side. So this is always labeled as number 1, then this is 2, this is 3, 4, 5, 6 and 7. Then this is generation. So generation 1, generation 2, generation 3 and generation 4. And if the two kids are having this A in between them, it means that they are identical twins. But if they are just connected like as 8 in Arabic, so it's like non-identical twins. If there is a double line instead of one line, it means cousin marriages or mating between the relatives. So this is how the whole pedigree has been organized. Analyze the pedigree to identify inheritance patterns related to dominant and recessive traits. So we have two different types of the traits. Dominant is the one that always express. It is always expressed, while the recessive one, it can skip generations. Okay, so this is how you learn as a hint about them. So walleye is a trait in swamp buffalo in which the iris is very light in color instead of the typical brown. And a single gene determines walleye trait in the swamp buffalo, and the allele for the eye is dominant to the allele for the brown eye. And then the pedigree is, so it means brown eye is a recessive one and walleye is dominant one. The pedigree below shows the lineage of these buffalo, and use the pedigree to answer question 36 and 37. So when it is walleye, it will be colored male, colored female, and brown, because it is recessive, so it will be empty. So it means that the disease is going to be looking down into question now. So gray one over here are unknown. If the walleye allele is designated as A and the brown eye is designated as small a, what is the genotype of the brother of individual 2? So individual 2 is this one. So this is the brother, right? This is the brother, and it's fully shaded, and it's wall eye. So if it is fully shaded and wall eye, so what it can be, we have to see that it can be capital A, capital A, small a, or capital A, small. So it can be both, right? It can be either capital A, or capital A, small a, because you don't know, because this wall eye is coming because of one capital A, right? So the next can be capital, or the next can be small. So it can be capital, or capital, small. Which of the individual indicates as having an unknown phenotype having a wall eye trait for the certain? So like where you are sure that it should have the trait. So who should be that one? How we can make a sure about 1, 2, 3, and 4? Okay, the thing is that you either look at, now this is 1, 2, 3, and 4, where you are 100% sure that this is the one with the trait. You need to look at their children. So we don't have children here, we don't have children here, we don't have children here, but here in number 1 we have children, and the children, this one is having capital A, but this one doesn't have the capital A, it only has small a, because this is blank. So this is small, and this is small, right? So here it must have a capital and a small, right? Or a capital and a capital. This is how it works. But of course it will have a capital and a small, because this individual is normal. So this is the one with capital A, small a. So it's number 1. Okay. Use the pedigree below, which shows the inheritance of Marfan syndrome in a family, to answer 38 and 39. So we have a pedigree, and in this pedigree they want you to identify the pattern of inheritance. Pattern of inheritance mean if it is dominant, if it is a dominant pedigree, or a recessive pedigree. This is what do we mean by pattern. And give one piece of evidence from the pedigree to support your identification. So always make sure that you provide the evidence from the pedigree itself. Look at the first generation. In the first generation we have only individual number 1 who is affected. But if you look at the children, if you look at their children, 1, 2, 3, 4, and 5, we have 5 children, and out of 5, 4 out of 5 are affected. It means that this is a dominant trait. This is one reason. Reason number 2, whenever these individuals, they got married, okay, and they should transfer the trait, but the possibility can be like, for example, over here you don't know what's happening, over here you still don't know like if they have it or no, but in general you will say that it is a dominant one. It's a dominant one, okay, because we don't clearly see that it skipped generations, because the skipping generation means the whole generation. But if you only look over here and you say that it skipped generation, no, it won't work, because the whole generation is starting from here and you go until here, okay. How you support your evidence? As I told you, the disease did not skip the generation, okay, so that's why it is dominant. And also it passes the trait from the parent to almost all the children, for example in from generation 1 to generation 2. And if it is recessive, you need to look for it skip the generation, and 2 normal parents give you a diseased one. We may see it comes down. Based on the pedigree, identify the probability that individual 38 marked by question mark will have Marfan syndrome, and draw a Punnett square to justify your identification. Okay. Now we need to identify the probability, like what's the probability that this individual goes over him. Okay, so starting from up, since this individual is normal, it means that this father is capital A small a, okay, and this is small a. Is that fine? Now it means that this individual this individual 3 is capital A small a, okay, and this is again small a. And the same goes over here. So we will make a Punnett square. Let's do a Punnett square. Capital A small a, small a small a. Okay, so small capital small. So it means that it's going to be 50%, because you can see over here the 2 are the normal one and 2 are the disease one. So capital A small a is equal to 2 out of 4. So probability is what, 50%, or 1 by 2 or 0.5. So this is the probability, how you say it, and we did the Punnett square already. Use the pedigree, which shows the inheritance of attached earlobes in the family, to answer the question 40 and 41. We have 2 normal, so this is called as skipping generation. Okay, this is called as skipping generation. And also 2 normal giving to an affected. Okay, 2 normal parents, 2 normal parents give birth to affected. Okay, so when you say the 2 normal give birth to affected one, it means that this is recessive inheritance. Okay, and once you know that it is recessive inheritance, what you can do after, that's how you justify it. So the trait skip the generation 1 doesn't have, but generation 2 has. And also 2 parents 1 and 2, they did not have the trait, but they gave birth to 2 1, 2 3 and 2 7, which are affected. Okay, and also over here parent 5 and 6, they didn't have it, but their children 3 has it. Based on the pedigree, identify the probability that the individual 2 2 is a carrier of the trait of the draw upon a square. So 2 2, so this is 2 2. We need to know, like, this individual is also a carrier. We need to check the probability. So simple is that the upper parents are carrier. So a same cross as we did in the previous. So capital A small a, and then small a. So it's a carrier. So carrier is this one, right? So heterozygous is a carrier, 50%, 1 by 2, 2 by 4, or 0.5. So anything you can write as your answer. That's great. We are done with 6.2. Now moving to 6.3. Non-Mendelian genetics. So this is where it doesn't follow the rule of the dominant and recessive. It has in between or blend ones as well. Okay, so first KPI, differentiate using examples and solving problems the patterns of inheritance of incomplete dominance and co-dominance. So when we say incomplete dominance is a blend. Okay, is a mixture, leads to a new trait. Okay, co-dominance you will say both express equally, both the alleles express equally. Okay, 2 pure breeding snapdragon plants were crossed, producing F1 generation. One had red flower and the other had the white flowers. The petal color is inherited by incomplete dominance. The predicted offspring phenotype percentage R. Okay, so producing the F1 generation. So when you produce the F1 generation, what you see, so this is incomplete, right? Red with white. When you do this cross, you know that you bring pink, and this is 100% pink, because this is F1, this is P. Okay, F1 leads to 100% pink, which is over here. This one is F2. When you cross 2 pink with pink, so pink crossed with pink, when you do this, you will get this percentage. What is the chance for a father who has a blood group AB and a mother who has a blood group O to have a child with blood group O? It's so simple. IA, IB, you do the Punnett square, right? Here we have I, here we have I. So it is IA and IA and IB and IB and I. So here IA and IB are dominant. So what's going to happen? This will lead to blood group A. This will lead to blood group A. This will lead to blood group B. And this will lead to blood group B. And the question says that what's the probability to have blood group O? It means 0%. We don't have any probability over here. But if they ask you what's the probability of A, it's 50%. What's the probability of B? It's 50%. Mackerel fish, the snail, the scale color trait under the control of the codominance when both, is written over here, where both the allele control the green and blue variants of the color scale. For a cross between a fish that has the scales with both green and blue color with another fish that has green scale. So blue and green. So it's like BG with only green, so it's GG. Okay, what is the probability that the offsprings will have the green scales? Okay, so we are going to do a Punnett square. We will do the Punnett square between the parents. So it's going to be like this. So BG and GG. So this is BG, this is BG, this is GG, and this is GG. And this GG is going to be what? Is going to be green. So what's the probability? 2 out of 4. That means 50%. So this is how you calculate it is 50%. A student crossed pure breeding of red flower Poinsettias with a pure breeding line white flower. So it's again red and white. The student observed that all the plants in the F1 have pink. We did this until here. Okay, F1 had pink. The student has then crossed the F1. So pink crossed with pink. I told you already what should come. So this is how we learn the things. So this is the answer. Search this answer down. So it is 25% ratio 50% ratio 25%. That means 1 ratio 2 ratio 1. But this 1 is red, 2 are pink, and 1 is white. So it is 1 2 1. So 1 red, 2 pink, and 1 white. This one. Use the diagram below to answer 46 and 47. The cross is made between a white body color and the chestnut body color, and we got a roan. So of course now roan is a mixture, right? So you will say that it's gonna be a mixture. So when it's a mixture, you got a totally different color. It's neither white nor chestnut, so it's going to be what? Incomplete dominance. But in some of the cases, like the same, if you have anything like this horse even, and it had stripes, like this is this coming from here, and the empty region coming from here, this empty is coming from there. So they have stripes, they have both colors together, they have the bands of the two colors. So this is called as a the codominance. So this will be codominance. Okay, so there are many examples of the incomplete dominance. Red and white gives you pink. Black and white gives you gray. And also over here, white and chestnut gives you roan. Explain your answer. So I already told you what's the pattern. Pattern is incomplete dominance. And how you explain the answer? The offsprings showed a mixture of the two colors, or roan is in between the chestnut and the white body color. Draw a Punnett square to show the probable results of the cross between a roan male horse and a white. So roan crossed with white. So roan is going to be, if it is chest, is represented by CC, this is WW, so this is CW. Okay, so CW crossed with WW. When you do this, So CW, WW, so CW, CW, WW, and W. Yeah, it should be W. So from here, you know that what's the ratio of the phenotype coming to the expected offsprings, you know. So that is 50%, 1:1, or 50%, or 2:2. Better not to say 2:2, because you know it's a mathematic rule. You need to simplify them. So make sure that you simplify. So this is the expected ratio that we get. 6.3.6 explain how X-linked genes are inherited. Include the analysis of pedigree. X-linked mean those who are present on the X chromosome. Now this X chromosome is present in both male and female. A male which will be XY. So any trait that's present on the X of the male, it is coming from in the male, the X is coming from mother, because Y is coming from the father. But if it is a female, so it is XX. So both parents provide one chromosome. Okay, use the pedigree to show the member exhibit an unknown disorder. Use it to answer 49 and 50. So we have this. So we have unaffected male and unaffected female, and we have affected male, affected female. Identify the most likely mode of inheritance of the disorder. More likely inheritance of this order. So it is how you will say this is X linked or sex linked, or the Y linked. So we will say it is X linked. Okay, so this is X linked. Why it is X linked? Because both male and female are affected. Okay, so you can say it in your own word. It's okay. And also you can say it is X-linked, right? There are more males than females, but both are affected. So this is how you say it. Identify the genotype of the affected female in the third generation. So since the generation numbers are not given, so this is number 1. Number 2, third generation, affected female in the third generation. And this female is coming from an affected father. It is coming from an affected father and a normal mother. But this normal mother is coming from this affected father. So this affected father gave the X from here, and the mother, which was normal, gave the X. So basically, this is normal, but the daughter is affected. Now, when you say daughter is affected, this daughter gain 1X from the mother, 1X from the father. And to be affected, both should carry a trait. So it means that this is a carrier. And this is X capital C and X small c. But over here, it is X small c and Y, because male only need one chromosome, right? So based on that, you can see this is affected. So it is small X small c and X small c. So you should know over here, sorry, X small c and X small c. I hope you have understood. If not, please rewind the video and try to listen again. What did I say? A woman is a carrier. Okay, so she is a carrier for the split hand trait and married to a normal man. So carrier mean one capital, one small. Use the Punnett square to find the probability of her first child is an affected boy. So carrier with normal. How we are gonna say that? Let's say it is X capital C X small c. So she is carrier, and she marries to a normal one, X small c and Y. Okay, because it's a man. We are not saying it is autosomal recessive. We will consider it as it is a sex-linked trait. So it's going to give you X capital, X small, X small, X small, and over here it is X capital C capital Y, X small c small y. Okay, so what is the probability that her first child is an affected boy? So over here we need to see this is a boy and this is a boy only, and this is normal because it has a capital C, and this is smaller because it has a capital C. So in that case, boy, the upper boy is capital C, right? So it's not gonna be affected, but the lower boy is a small c. So X small c and Y is only 1 out of 4 or 25%. So this is the probability that you need to say. Use the recombination data to map the distance between the genes in the chromosome. This is one of the easiest but tricky questions that can give you like quick marks. So choose the correct diagram that represents the relative positions of the 4 genes over here. So it's a set of the genes that A, B, C, D and how they are there. So find the longest one, the biggest one, which has the maximum percentage of the crossing over. So maximum percentage is over here, A to B. So when you say it is A to B, so A should be on one end and B should be on the other end. This is A, D, no. This is no. This is A, this is B, yes. And there is no other, so it's quite obvious. I'm not gonna look anything else. Use the table below that shows the recombination frequencies of the 4 genes. Okay, and to answer question 53 and 54, again, look for the longest frequency. Okay, construct a genetic map. So you can construct, so draw a line and at one end put A and at another end put B, because this is the longest one, it's 37. Next, who is closer to A or further from A? So C. If this was 37, so it's going to be here somewhere. So it will be 23 and it will be 14. Okay, now C to D is 15, but B to D is 29. So we will go this side. Okay, so this is 15, this is D. So that's how it works. And B to D, it will automatically become 15 plus 14 is equal to 29. So this is the answer. The sequence is A, D, C, B. Identify the 2 genes that have the highest chance of undergo recombination during the meiosis and explain your answer. Those who are farthest apart, they have the highest chance to undergo recombination. But those who are very close to each other, they have the least chance. Okay, so here who has the highest chance? It is A and B because they are farthest from each other. This is the reason. The distance is the greatest. But who has the smallest? So look, how you find the smallest now? I know you are gonna look into the table. When you look into the table, so what you say over here, who is the smallest? Oh yes, this is the smallest. So B and C will go together. Is that correct? Of course no, because we still don't know how much is this and we need to calculate. Look, this is 29 and this is 37. So 37 minus 29 is gonna be 8. So A to D, which is not given over here, A and D is 8. So the one who has the highest chance is A and B. If we say lowest chance, then it will become A and D. So sometime you don't look only into the table, you look into this map as well. Like if there is anything missing, you fill out. Describe how mitochondrial and chloroplast genes are inherited. So mitochondrial genes mainly inherited from the mother, and the same goes for the chloroplast in the plants. A scientist is studying the inheritance of genetic disorder that affects energy production in the human cell. This is related to mitochondria, of course. Okay, so you can easily choose because this is related to the energy production. The disorder is passed down only from affected mothers to all their offsprings, while affected fathers do not pass the trait. Please remember this one. Whenever you see that there is an affected male, so if you see a pedigree, I don't know if they have given a pedigree. No, they did not give a pedigree. So if you see a pedigree and that pedigree shows you, for example, like this, sorry, so for example, like this is an affected male and this is a normal female. So what you expect down, like there should be someone affected at least. So you have like 6 children, okay, and none of them is affected. So it means that the father did not pass on the trait, did not pass on the disease. It means that it is a mitochondrial trait. So disorder is caused by mutation in autosomal gene? No, because we are talking about the energy. The disorder is caused by mutation in the mitochondrial DNA? Yes, and it is inherited exclusively from the mother? Yes. Disorder is caused by the mutation in Y-linked? No. Disorder is caused by mutation in chloroplast? No, because we are talking about energy production. Explain using examples how environmental factors influence gene expression and can lead to phenotypic plasticity. So you should know that the environment plays a major role in the gene expression. How come? This is because the phenotype is equal to genotype plus environment. So environment plays a major role in the phenotype. Okay, newborn white sheep and spell sheep are two breeds of the sheep that are commonly rare in Norway. And body mass at weaning, which is the time when the baby sheep stop drinking their mother's milk, is an important to measure the sheep farmers. Scientists measure the dry mass at the weaning in the NWS and the spell from faulty flock and sheep distributed around again. And the table below shows the mean body mass of the two breeds. The weather was very different between the 2 years. Daily temperature average is 1.9 degree colder in 1990, and there was 4% less participation in 1990. Thus, the 2 years can be considered different environments. Use this information in the table to answer questions 56 and 57. So we have year 1989, 1990. So this is a 2 year span. The mass for the new Norwegian white sheep, and this is for the spell mass. So you can see there is a difference in the mass, right? So 44 and 40 and 44 and 42. On average, determine how much larger spell bred sheep were at weighing 1990 compared to 1989. So just take the difference between them. Okay, so in 1989, so this is 40. So what they want, sorry, how much larger spell bred sheep were at weaning in 1990 compared to the 1989. So just take the difference between them. It should be spell mass, so it is 42.9 minus 40.8. 9 minus 8 is 1, so 2.1, so 2.1 kg. Okay, write a conclusion based on the results. So of course, the topic that you are studying, it means that the mass increases under colder environmental conditions. So this is how you say the conclusion. Hydrangea plants of the same genotype are planted in a large flower garden. Some of the plants produces blue flowers and the other produces pink flower. This can be best explained by which of the following? Of course, they have environmental impact, but we will see what kind of environmental impact. Completely dominant? No. Co-dominant? No. The fact that a mutation? No. So it means environmental factor. So this is the answer. Which of the following is not an outcome of the environment modifying phenotype? The changing of the color of an animal's fur as the temperature change, it is one of the environmental factor that we see. We need to see not. This is the one that happens. The increased intelligence attending the school for many years, yes, that can happen. The very short stature of a kind of a tree that grows at a high altitude compared to the same kind growing at low altitude? Yes, that can be environmental factor. The pink flower snapdragon that result from a cross between red and white? No, this is incomplete dominance. This is incomplete dominance, so this is not happening, and this is your answer, right? So this is how you work on it. Okay, moving to the last part of chapter 6, 6.4, chromosomal inheritance. Explain by examples the human genetic disorder can be a result of inheriting single gene, recessively inherited disorder, and dominantly inherited disorder. So basically, chromosomal inheritance depends on the changes in the gene or changes in the chromosome. So we need to see like what are the changes in the genes and chromosomes. Okay, PKU, phenylketonuria, is an autosomal recessive genetic disorder in mammals. So it means that it will happen because of small a, let's say. PKU is caused by a mutation in the enzyme. So when there is a mutation in the enzyme, it is a gene mutation. Okay, clear. By a change in one nucleotide. Okay, we know that it's change in one nucleotide. This enzyme is responsible for converting phenylalanine to tyrosine. An individual with a mutated enzyme accumulate toxic levels of the phenylalanine in the blood. Use this information and your knowledge to answer questions 60 to 62. Identify the PKU is a disease caused by a gene mutation or a chromosomal mutation. So of course it is a gene mutation because it is happening because of the mutation in the enzyme in one nucleotide. Use your knowledge of PKU to describe is this a recessive or a dominant trait. Since we say that it is an autosomal recessive, yeah, so it's written up, so it is a recessive trait. PKU is considered as a recessive disorder. Then what is the probability of the two carriers? So of course to have a normal child. So two carriers to have a normal, it will be capital A small a crossed with capital A small a. So normal will be capital A, so it is 25%. Okay. Which of the following is a human disorder that is caused by a dominant allele? So you should know like which one is dominant. So you studied 4. Only Huntington was dominant. Remaining Tay-Sachs, phenylketonuria, and cystic fibrosis, all of these are recessive. Which of the following is correct about PKU? We know that it is recessive, okay, and it is a genetic, it happens because of the gene mutation. Let's see what they want to ask from down. It is caused by the many gene mutation. Many gene mutation? No, we said it's a mutation in one gene. It results in the production of an impaired form of enzyme. Yes, we studied this, phenylalanine to tyrosine. It can be inherited as a dominant. No, it's a recessive trait. It is caused by an extra chromosome. No, it is caused by a genetic disorder. So B. Relate non-disjunction disorder to both autosomes and sex chromosome. It will lead to Down syndrome, Klinefelter syndrome, and Turner syndrome. So first of all, what do we mean by non-disjunction? Non-disjunction is a process in which chromosomes cannot separate, okay, and lead to an extra or a less chromosome. Okay, so for example, in Down syndrome you have an extra, so it is 47. Klinefelter syndrome you have an extra, it is 47. And Turner syndrome you have a less, so this is 45. And how you state them, this one is n plus 1, this one is n plus 1, and this one is n minus 1. Okay, use the diagram which shows a summary of the process of meiosis in human to answer question 50 65 and 67, 65 till 67. So you have 46. It's me after meiosis 1 you should get 23, but you got 24 here. So what will happen now? Identify the name of the process that caused this one. Of course, this is non-disjunction, right? This is non-disjunction. and describe how it occurs. Simply you need to say that it's a failure of the separation of the homologous chromosomes. So cannot separate or fail to separate, but the homologous chromosomes, they fail. Non-disjunction lead to failure of the homologous chromosomes to separate. Predict the result of the gamete W. So this is gamete W fertilized with the normal gamete. So from 24, it is 24. The normal should be what? So 24 plus 23 should be normal. So it's going to be having 47, right? So the resulting zygote will have an extra chromosome, and it will be abnormal. Identify the name of the disorder that is caused by an extra copy of the chromosome 21. So chromosome number 21, it is Down syndrome. Okay, but if you have a less chromosome in the sex chromosomes, then it will be Turner syndrome, this one. So it is like X0. If you have an extra X chromosome, this will be XXY. Okay, the law. Okay, use the diagram below to answer 6870. This is what I was saying, you an extra XXY, so you should know, like, this is Klinefelter. Okay, identify the abnormality. We said it already, this is a Klinefelter syndrome or extra chromosome. Identify the name of the error that occurred. So it is again non-disjunction. Okay, so this is non-disjunction. Identify the name of the disorder. Oh, so this one will come here, but identify the abnormality. It is an extra chromosome, so you will say it is an extra sex chromosome. Extra sex chromosome. Moving to the next one. 4 students provided the cause of the 3 genetic disorders. Who gave the correct answer? Okay, where we can see, target anyone. Which one is easy for you? Let's say Down syndrome. Okay, extra sex chromosome, no. Extra autosome, yes. Missing sex chromosome, no. Extra autosome, yes. So it's either 1 or 4. Now Klinefelter, missing sex chromosome, no. So it means now 1 is not there, so it is 4. So 4 is the answer, and this one. So it is extra sex chromosome XXY, missing sex chromosome X0, and extra autosome at 21 chromosome number. And that's it. We reached to end of topic 6. Inshallah, see you soon with topic 7. Thank you.